Now let's discuss about the eukaryotic chromosomal structure and compaction. Our DNA measures about 2 meters and 2 nanometers in diameter. This must be firmly packed to fit in the cell nucleus. Does anyone know what is the process of DNA forming into chromosomes? Thank you for the answer. This starts with a double helix DNA. Then, the DNA wrapped around histones. The combined tight loop of DNA and protein is the chromosome. Multiple chromosomes are coiled together and these then stuck on top of each other. The end result is a fiber of packed nucleosomes known as a chromatin. The DNA is tightly packed into the familiar structure we can see through a microscope, which is the chromosomes. Another question: how many chromosomes does our body have? Let's go to the chromosomal theory of inheritance. The discoverer's discovery. First, we have Theodore Boveri in 1902 observed that the proper embryonic development of sea urchin does not occur unless chromosomes are present. Also in the same year, Walter Sutton observed the separation of chromosomes into daughter cells during meiosis. And these two observations lead to the development of the chromosomal theory of inheritance. Fast forward to 1910, Thomas Hunt Morgan started his work with Drosophila melanogaster, a fruit fly. He provided experimental evidence to support the chromosomal theory of inheritance. During that time, he already knew that big letter X and big letter Y have to do with gender. He utilizes normal flies with red eyes and mutated flies with white eyes and crossbreed them. With the Drosophila melanogaster, the wild-type eye color is red, which is the big letter X and W, and is dominant to white eye color, which is the big letter X and small letter W. Using these genotypes, he was able to conclude that the gene for eye color was on the X chromosomes. This trait was determined to be X-linked and was the first X-linked trait to be identified. Males are said to be homozygous, in that they have only one allele for any X-linked characteristics. So, what does Gregor Mendel have to do with the chromosomal theory of inheritance? The cell division cycle. So, does anyone know what are the stages of a cell cycle? Thank you for the answer. So in cell cycle, the stages are G1, S, G2, and M, including the cytokinesis. G1 is the period after cell division and before the start of DNA replication. Cells grow and monitor their environment to determine whether they should initiate another round of cell division. In S, it is the period of DNA synthesis where cells replicate their chromosomes. And in M's stage, is the actual period of cell division consisting of prophase, metaphase, anaphase, telophase, and cytokinesis. By the way, the G1, S, and G2 is part of the interphase. Mitosis ensures that every cell in an organism carries the same chromosomes. Stages of mitosis. As I've mentioned earlier, we have the prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. In prophase, chromosomes condense and become visible. Spindle fibers emerge from the centrosomes, nuclear envelope breaks down, and nucleolus disappears. Prometaphase. Centrosomes continue to condense, kinetochores appear at the centromeres, mitotic spindle microtubules attach to kinetochores, and centrosomes move towards opposite poles. Metaphase. Meiotic spindle is fully developed. Centrosomes are at the opposite poles of the cell. Centrosomes are lined up at the metaphase plate, and each sister chromatid is attached to a spindle fiber originating from opposite poles. Anaphase. Cohesin proteins binding the sister chromatids together break down. Sister chromatids, now called chromosomes, are pulled towards opposite poles, and non-kinetochore spindle fibers lengthen, elongating the cell. Telophase. Chromosomes arrive at opposite poles and begin to decondense. Nuclear envelope material surrounds each set of chromosomes, and the meiotic spindle breaks down. Cytokinesis. In animal cells, a cleavage furrow separates the daughter cells, and in plant cells, a cell plate separates the daughter cells. The control of cell cycle. The three major checkpoints are the end of G1, the end of G2, and during metaphase of M phase. G1 checkpoints check for nutrients, growth factors, and DNA damage. G2 checkpoint check for cell size and DNA replication. And metaphase checkpoint check for chromosome spindle attachment. Let's watch this video about the cell cycle checkpoints and regulations animation.
Cell cycle. The cell cycle has three states, which are quiescent, interphase, and mitosis, and 5 phases, G0, G1, S, G2, and M. The quiescent state, also known as the senescent state, comprises of the G0 phase. Examples: neurons, cardiac muscle, and RBCs. Interphase state consists of G1, S, and G2 phases. And the mitosis state consists of the M phase. Prophase, metaphase, anaphase, telophase occur during this phase. The three major cell cycle checkpoints are the end of G1, the end of G2, and during metaphase of the M phase. Progression through checkpoints is regulated by cyclins, cyclin-dependent kinases, and tumor suppressors. During the G1 phase, cell cycle progression is controlled by a variety of different cellular mechanisms, such as tumor suppressors, genes that code for transcription factors, proteins that detect DNA damage, enzymes that allow the cell to progress to the S phase, and signaling molecules that activate those enzymes. The gene E2F regulates expression of transcription genes and cyclin-dependent kinase 2, cyclin E, and cyclin A. If there is significant DNA damage, P53 stimulates production of P21. P21 binds and inhibits all cyclin CDK complexes, which leads to arrest of cell cycle until the DNA damage is repaired and P21 levels drop. The tumor suppressor retinoblastoma protein inhibits E2F expression. Cyclin D CDK4 and cyclin D CDK6 complexes phosphorylate retinoblastoma protein that leads to inactivation of retinoblastoma protein and expression of E2F. The expression of E2F then leads to the expression of transcription genes and formation of cyclin E CDK2. Therefore, the cell is pushed into the S phase. Cell cycle regulation in S and M phase. Cyclin A CDK2 is required for DNA synthesis. Cyclin A CDK1 and cyclin B CDK1 promote the events of mitosis. Towards the end of mitosis, the anaphase promoting complex causes ubiquitination and destruction of cyclin A CDK1 and cyclin B CDK1 that leads to termination of M phase with anaphase, telophase, and cytokinesis. The most radiosensitive phases are the G2 phase and mitosis. The least radiosensitive phase is the latter part of the S phase.
And the last is the stages of meiosis 1 and 2. Let's head first to the stages of prophase. We have 5 stages of prophase. And first, we have leptotene, which is the replicative chromosomes condense. And second, zygotene, synapsis begins. And in pachytene, the third stage, a bivalent has formed and crossing over has occurred. In the fourth, diploidine, synaptonymal complex disassociates. And in the last, diakinesis, it is the end of prophase 1. And finally, the meiosis 1 and 2. It is just like the mitosis but doing its process 2 times. So, the stages of meiosis 1 and 2 is the prophase 1, metaphase 1, anaphase 1, telophase 1, and the prophase 2, metaphase 2, anaphase 2, and telophase 2. And cytokinesis also is included. For more information and further clarification, just check out our references.